Okay, so continuing from part one, this is part two of the chapter 6 lecture on viruses. And so we were talking about the general structure of viruses, and we have some atypical viruses, the pox viruses, and some of the bacteriophages. The pox viruses don't have a typical capsid, and they are covered by a dense layer of lipoproteins instead of a protein capsid. Then some of the bacteriophages are just viruses that infect bacterial cells. They can have a polyhedral nucleocapsid that also includes a helical or spiral shaped tail with attachment fibers. And the the bacteriophage that I was talking about in the previous slide, which is the polyhedral nucleocapsid with the helical tail and attachment fibers, which is here. To me, that is what you will see typically when you have cartoon viruses. That's what that looks like. It kind of looks like a spider. But you have the nucleocapsid, that's the polyhedral, and then you have these attachment fibers. Um, this one here is a pox virus, and you can see, that it has this, core membrane. It has an outer envelope. It has this other lateral body layer, some soluble protein antigens. You've got your, of course, nucleic acid core. So it has a really different structure to the other viruses that we've talked about. This is an electron microscope view of a bacteriophage. I meant to say that this is a bacteriophage that infects E. coli bacteria. Okay, so basic types of viral morphology. This is from a table, or figure 6.10 in your book, and it's just showing some different types of complex viruses. And you have this is the outside of a pox virus. This is your bacteriophage with the flexible tail. These are your enveloped viruses, the helical enveloped virus, which is going to be, this is the mumps virus. And then this one is the rhabdovirus. And then these ones over here are ones that have an icosahedral nucleocapsid. And this is a herpes virus. If I could get it to highlight, this is herpes virus. And then this one is the HIV or AIDS virus. So those are different enveloped viruses there. Then there are some non-enveloped or naked viruses. This is a helical virus. This is the plum pox virus with that. These are the capsimers of a helical capsid, so protein coat. And then you have this one. These are some naked or non-enveloped viruses that are icosahedral in shape. And this is the poliovirus here and the papillomavirus here. Okay, so we're going to carry on. We're talking about the general structure of viruses, and now we're going to talk about the nucleic acids. And so the viral genome, as I said at the very beginning of the lecture, can be either DNA or RNA, but will not include both DNA and RNA. The viral genome contains the genes necessary to invade a host cell and take over the host infrastructure to make new viruses. The number of genes varies from one virus to the next, and there can be just a few to hundreds. So with DNA viruses, they're usually double-stranded. That's what this DS represents, but they can be single-stranded, or SS. They can also be circular, like the ones in bacteria, or linear, like the ones in the eukaryotes. RNA viruses are almost always single-stranded. They can be double-stranded, and they may have different RNA pieces or segments. that contain the genome. Single-stranded RNA genomes are ready for translation into proteins, and they are called positive sense RNA. And then there are single-stranded RNA genomes that need to be converted into the proper form for translation, and those are negative sense RNA. So in order for the virus to replicate or duplicate, there are some preformed enzymes that need to be present, and those include polymerases, which are responsible for synthesizing DNA or RNA, replicases, which copy RNA, and then another enzyme called reverse transcriptase, which is used to synthesize or make DNA using an RNA template. And an example of a virus that uses the reverse transcriptase is the HIV virus. And the reason that these enzymes are necessary to be preformed is because viruses they don't have the equipment to make their own enzymes. They don't have their equipment to synthesize DNA themselves. And there are even some viruses, interestingly enough, that can borrow or take things from the host cell. For example, arenaviruses can actually take ribosomes from a host cell, and then retroviruses can use the tRNA molecules from a host cell. So even though they can't do it themselves, there's very little impact on the virus. They've figured out how to take over the host infrastructure to do all the things that they need to do. So now we're going to move into section 6.3 and talk about the way that viruses are classified. So the main things that are used to classify viruses are the structure, chemical composition, and genetic makeup. So there is, according to the International Committee on the Taxonomy of Viruses, there are 7 orders, 104 families, and 505 genera, which is plural for genus, of viruses. As far as the nomenclature goes, the family name will be italicized and given the suffix viridae. So if you see a virus name ending in viridae, that indicates that it's a family name. And a genus name is also going to be italicized and have the suffix virus. So that's the way that you can tell a family name from a genus name or genera. Okay, so viral species. There are distinct virus types that have certain properties or characteristics in common. And those things could include things like host range, which is the variety of hosts that particular group of viruses can use. Pathogenicity, like the level of pathogenicity that a group of viruses possesses, and then also the genetic makeup. So the characteristics for inclusion within a virus family include the type of capsid that they have, the number of nucleotides in a nucleic acid strand, the presence or lack of and type of envelope, the overall size of the virus, and the area of the host cell that the virus multiplies in. Also, the microscopic morphology or appearance of a virus can be used to classify viruses. The anatomical or geographic areas that are inhabited can be used to classify viruses, and then the effects of a virus on the host are used to classify viruses. So then you also will have acronyms that blend more than one characteristic of a virus in order to kind of be descriptive of some of the characteristics of the virus. Table 6.2a in your book has some important virus families in terms of ones that impact humans. It includes the family, the genus, the common names, and then also the name of the disease that those particular viruses cause. So now we're going to talk about animal viruses and the modes of multiplication. We're going to talk about the virus-host relationship, and then we're going to relate the stages in the multiplication cycle of animal viruses and talk about what's happening at each stage. In addition, we're going to look at how 3 ways that animal viruses enter host cells. We're going to define replication as it relates to viruses, and then we will be explaining the ways in which animal viruses are released by a host cell, and then describe the cytopathic effects of viruses and the possible results of persistent viral infection. So the general phases in animal virus multiplication include adsorption, which is just when the virus binds to the host cell, penetration, which is when the genome is injected into the host cell or when the genome enters the host cell, uncoding, which is when the viral nucleic acid is released from the protein capsid, synthesis, which is when viral components are produced. So synthesis, making the virus is made, and synthesis. Assembly is when those components are put together, when new viral particles or viral bodies are put together. And then release is when the assembled viruses are released from the host cell by exocytosis or cell lysis. And so there is a figure in your book. It's kind of hard to see on here, but it has the different stages or phases of the multiplication cycle numbered with pictures. So it has adsorption, it's attaching, penetration, which is when the genome is injected into the host cell, uncoding, which is when the capsid comes off of the viral genome, synthesis, which is when the parts of the virus are being created, assembly, which is when those parts are being put together, and then release, which is when either the virus is released through exocytosis or the cell lyses to release the viral bodies. Okay, so adsorption and host range. So the way that a virus, finds a host cell is by coincidence. It collides with a susceptible host cell and then adsorbs or attaches to that cell. There are usually going to be receptor sites on the host cell membrane, and the range of the hosts, or the variety or spectrum of the cells that a virus is able to infect, that's called the host range. So if you have a certain family of animals that a particular virus can infect, that family of animals would be the host range. And here are some examples. Hepatitis B infects human liver cells. Poliovirus infects primate intestinal and nerve cells. Rabies infects different cells from many different mammal species. Okay, so penetration and uncoding. Animal viruses have to penetrate the cell membrane of the host and get that nucleic acid into the interior of the host cell in order to be successful. The 2 most common ways that a virus can penetrate a host cell are by fusion or endocytosis. Fusion is in enveloped viruses. The viral envelope fuses with the host cell membrane by rearranging those lipids or phospholipids there. And then also there's endocytosis, which we talked about when we talked about cell transport. And enveloped or naked viruses can get their nucleic acids into the host cell by the mode of endocytosis. So the entire virus will be engulfed by the host cell and enclosed in some type of vacuole or vesicle. Then once the virus is inside of the host cell, the uncoding happens and the nucleocapsid or nucleic acid is released into the host cell. Okay, so I'm going to stop here. There's a figure that shows the 2 modes of viral penetration, and we'll do